Hi students, in the last class we discussed about the properties of monosaccharides, and coming to this class we are discussing about oligosaccharides. See, we already discussed about oligosaccharides, means the definition for oligosaccharides is 2 to 10 monosaccharides are there. See, oligosaccharides, there is a combination of monosaccharides which are having 2 to 10 monosaccharides which are linked by the glycosidic bond, right? So these are called as oligosaccharides. So within that oligosaccharides there is an disaccharides are there, trisaccharides are there, tetrasaccharides are there, right? Penta Saccharides are there. Right. So like this here, based on that number of monosaccharides, we are giving the names to the oligosaccharides. So how these are linked, we already discussed regarding to this point also in the last class. See, whatever that monosaccharides are there, the monosaccharides are linked by the glycosidic bond. The monosaccharides are linked by the glycosidic bond. So which are forms the glycosidic linkage? Then these are forms the oligosaccharides, different types of oligosaccharides. So the very first one is the disaccharides. Disaccharides means which are having 2 monosaccharides. The glycosidic bond is 1. The number of glycosidic bonds are here 1. So examples for the disaccharides, there is an maltose is there. See, very simple. Maltose is the disaccharide having glucose and glucose. It's having that linkage, yeah, the repeated units or the components are here, glucose and glucose. Maltose is the disaccharide, it's having 2 glucose molecules. The linkage is here alpha 1,4 linkage, alpha 1,4 linkage. So how the maltoses are formed? We know about the starch. Starch is broken down, right? Starch is broken down. We know we are eating the food contains the starch. Within that mouth there is an alpha salivary amylase enzyme is there. That enzyme involved in the cleavage of starch. So during the cleavage of starch there is an dextrins are produced. Dextrins are produced. These are the intermediate molecules. Again the dextrins are cleaved, which forms the monosaccharides, and the next one is the disaccharides. So the disaccharide units are nothing but maltose. The maltoses are formed. So starch is broken down into that some of the dextrin molecules. Again the dextrins are cleaved into the monosaccharides and the disaccharides, right? The disaccharide example is here maltose. So here the glucose and the glucose molecule. We already discussed the difference between that alpha 1,4 linkage and the beta 1,4 linkage. Here there is an alpha 1,4 linkage is present. This one is also called as malt sugar. This one is also called as malt sugar. Remember, whatever that fermentation process is there, see, we know about that alcohol production. We know about the beer production, right? Within the alcohol, beverages production. Within the beverages productions, we are using that malt sugars. Whatever that cereals are there, the cereals are subjecting for that breakdown process. Finally, okay, they get that malt. Malt is nothing but maltose. Then that maltose is subjecting for the fermentation process. This is one of the raw material for the preparation of beers, for the preparation of alcohol, for the preparation of wines and different kinds of products. So here this is the malt sugar, right? The malt sugar is nothing but maltose. Here the linkage is alpha 1,4 linkage, right? That is about the malt sugar. Coming to the next one is the sucrose. Here we know about the sucrose because most of the plants, okay, within that plants, the transport form of carbohydrate. Within the plants, the transport form of carbohydrate is the sucrose. All of you remember storage form of carbohydrates in plants is starch. Storage form of carbohydrates in plants is starch. Transport form is the sucrose. Transport form is the sucrose. See, we know about that insect pollination process. During the insect pollination, the insects are attracted by the colored flowers. So that is the one case, and second one is the secretion of nectar. That is nothing but honey production, right? So here, that honey especially contains the sucrose. That honey contains the sucrose. So here, the sucrose is the disaccharide, right? It's having the components are glucose and the fructose. It's having the components are glucose and the fructose. There is an alpha 1 and beta 2 linkages are there. There is an alpha 1 and beta 2 linkages are there, right? See, this one is called as cane sugar. This one is called as cane sugar. So why this one is called as cane sugar? Because whatever the sugar cane we know, right? If we crush the sugar cane, we get the sugar cane juice, right? This one is also called cane juice. That cane juice contains sugar is the sucrose. That cane juice or the sugar cane, okay, that one contains sugar is the sucrose. This one is also called table sugar. What we are using at house for the making coffee or tea, right? Different types of sweet substances. We are using that sugar, right? That one is nothing but table sugar. So sucrose is also called as table sugar. Because that the cane sugar subjecting for that preparation process, finally we get the sugar. That's the reason why this one is called as cane sugar, and this one is also called table sugar. Here the shape is that, right? So that's the reason why here this one is called as table sugar. And this one is the non-reducing sugar. Non-reducing sugar means the sugars are not react with any other components. See, whatever that sucrose is there, within that sucrose there is a glucose and the fructose is there. Okay, both the functional groups are linked. Both the functional groups are linked here. Alpha 1 and beta 2. Alpha 1 and beta 2 linkages. See, within the glucose, first position there is an aldehyde group is present. Within the fructose, at the second position there is an keto group is present. So both these are linked. Both these are linked. The linkage is called as alpha 1 beta 2 linkage, right? So that's the reason why there is no free function groups. So that is the reason why the sucrose is unable to react with any other reagent or any other components. So these are inert, which are unable to react with the other components, right? So this one is called as non-reducing sugar. The sucrose is called as non-reducing sugar. And coming to the next one is the lactose. We know, I thought I'm expecting we know about the lactose. Whatever that milk contains sugar is the sugar is called as lactose. The sugar is called as lactose, right? So here there is a glucose and galactose components are present. Within that lactose there is a glucose and galactose components are there. Here linkage is the beta 1,4 linkage. The linkage is here beta 1,4 linkage. This one is also called milk sugar. Lactose is also called milk sugar. All of you remember the lactose, the word indicates here, very familiar, right? Here the lactose is the sugar, it's having the glucose and the galactose, right? Next one is it's having that beta 1,4 linkage, and this one is called as milk sugar, right? This one is called as milk sugar. All of you remember this is the least sweet taste sugar. This is the least sweet taste sugar. The total carbohydrates, the least sweet state sugar is the lactose, right? So that is about the lactose. And last one is that Trihalos. Trihalos, this is one kind of the disaccharide. It's having that glucose and glucose. Here linkage is alpha 1,1 linkage. This is the quite different one. Okay, first group aldehyde group. Again first group aldehyde group. Both the functional groups are linked. So both the functional groups are linked. There is no free functional groups which are unable to react with the other components. That's the reason why the Trihalos is also non-reducing sugar. The Trihalos is also non-reducing sugar. I'll let you remember whatever that insects are there, the insects contains that lymph, okay, hemolymph of insects. Especially this one is present in the hemolymph of insects. The Trihalos that is present within that insects. Okay, the insects contains that hemolymph is there. Within that hemolymph, the Trihalos is present, right? So that is about that the disaccharides, maltose, sucrose, lactose, and the Trihalos. So that is about the different names of the disaccharides also, right? And coming to the next one is the trisaccharides. See here, the tri means here 3 monosaccharides are there. The very first one is here the fructose, right? Here the very first one is the fructose, and next one is the glucose. Last one is galactose. So there is an trisaccharides are there. Within the trisaccharides there is an fructose is there, glucose is there, galactose is there. So the fructose, glucose, galactose, these three are combined forms the raffinose. The sugar is called as trisaccharide. Name of the trisaccharide is raffinose. And coming to the next one is the tetrasaccharides. Here fructose is there, glucose is there, galactose and galactose. Fructose is there, glucose is there, galactose and galactose. Here name of the sugar is the stachyose. So here there is in trisaccharides and tetrasaccharides are there. Here raffinose and the stachyose. These are the best examples. Remember the composition here are only different with here fructose is there. Here also fructose is there. Here glucose is there. Here also glucose is there. Here galactose is there. Here also galactose is there. Okay, only one additional galactose molecule is present. Right? Here number of glycosidic bonds are 2. Here number of glycosidic bonds are 3. So the raffinose and stachyose which are present in the phloem, whatever that phloem, okay, the sieve cells are there. Within that phloem there is an sieve cells are there. For that the phloem formation, especially raffinose and the stachyose, both these are supporting. Right? And coming to the next one is the pentasaccharides. Here the pentasaccharides means 5 monosaccharides are there. Same thing. Here there is an fructose is there, glucose is there, galactose, additional galactose molecule is there. This one is called as verbacose. See the verbacose seen in sieve tubes. See the sieve tubes are only seen in angiosperms. See, phloem is present in all the plants. Here, this one is also related to the phloem, but the sieve tubes only present in angiosperms. So, the simple explanation for the trisaccharides, tetrasaccharides, pentasaccharides. See, almost the components are same, but number of components are different. Here 3, here 4, and here 5, right? Here the fructose, glucose, galactose. Here fructose, glucose, galactose, no doubt about it. One more, additional galactose is present. The same here, fructose, glucose, galactose. Again, galactose. Again, one more additional galactose is there. So that is the pentasaccharide. We have to remember the sequences of these tri, tetra and pentasaccharides, and names also very important. The examples also given in the examination. So here the raffinose, stachyose, which are present in the phloem. Verbacose is different and this is advanced. That's the reason why this one is present in the higher plants. Okay, that is about that oligosaccharides. In the next class we will discuss about the heteropolysaccharides. Coming to the next topic is the polysaccharides. We know polysaccharides means many number of monosaccharides are combined by the glycosidic bond, and which forms the polysaccharide structures. These are also huge structures, right? See again here two types of polysaccharides are there. If these are made up of with single type of sugar or same sugar, which are called as Homo Polysaccharide. Another one is the which are made up of with more than one type of sugar. These are called as heteropolysaccharides. So here the concept is very quite easy. Homo and heteropolysaccharides. Homo polysaccharides means which are made up of with a single type of sugar or the same type of sugar. Heteropolysaccharides which are made up of with more than one type of sugar, right? See again here we have to discuss here about that Homo polysaccharides, means types of Homo polysaccharides and types of heteropolysaccharides. Here there is an Homo polysaccharides are there. In that case there is a cellulose is there, starch is there, right? Glycogen is there, right? And next one is the chitin is there, right? All these are the best examples for the Homo polysaccharides. And one by one we will discuss about Homo polysaccharides and the properties of the Homo polysaccharides. See Homo polysaccharides, and the very first one is that cellulose. if okay, when we heard the cellulose name, then immediately in our brain the plant cells are remembered. We easily remember the plant cell because the plant cell walls are made up of that cellulose, right? Major portion is the cellulose, especially whatever the primary cell wall is present, right? So here the cellulose is the Homo Polysaccharide made up of with 6,000 to 10,000 molecules. See, sir, what are the molecules? Glucose molecules. Here repeated, okay, the cellulose is made up of with repeated units of glucose molecules, repeated units of the glucose molecules. So many number of glucose molecules, right? Especially the number is 6,000 to 10,000 molecules of glucose units, okay, right? Which are linked by the glycosidic bond, So what type of glycosidic bond is seen in the cellulose? Remember, within the cellulose there is an beta 1,4 linkage is present. Last time I already drawn. This is the one of the structure. Okay, this is one of the cyclic structure of the glucose, right? So this structure is called as beta 1,4 linkage. This one is called as beta 1,4 linkage. So this is the beta 1,4 linkage. That beta 1,4 linkage is seen in the cellulose structure, and the very important point of the cellulose is the linear structure. Cellulose is always forms the linear structure like this. There is no branches. Okay, if the branches are present like this. So within the cellulose, the branches are absent. So here cellulose forms the linear structure only, branches are absent, right? And coming to the next one is within the plant cells there is in 40% of the cellulose is present. Whatever that plant cells we have taken, whatever that cell wall we have taken, right, within that cell wall content there is in 40% of the cellulose is present. So the cell wall content total is 100%, within that 40% is cellulose is there, right, and remaining hemicellulose, pectin, superin, all these are also present. All the component percentage is the 60, the only cellulose percentage is the 40, right? So that is about that plant cell wall, right? And 90%, whatever that cotton, we know about that cotton, right? Cotton fibers are there, which are very fair, color, means very pure white color, because see, these are purely made up of with cellulose only. See, maybe the cell wall content or other contents may be mixed with the other components, but whatever that cotton fibers, the fibers are very pure in color, okay, especially white color, right, because of these are purely made up of with the cellulose. So the percentage of the cellulose is 90% within the cotton, right? And tunica, what are some of the animals are there? I'm not talking about insects here, I'm talking about animals. Some of the animals are there. Within the animals, okay, whatever that covering skeleton is present, the covering skeleton is made up of with the cellulose. So tunica, exoskeleton, that is nothing but exoskeleton. Especially in some animals, the tunica formation done by the cellulose structure. And the cellulose is a very important component for the paper industry. Because what we are writing, that white papers are there, the white papers especially made up of with that cellulose components. So the cellulose components we are extracting from the plants. Okay, not we are extracting, the paper industry extracting from the plants. So the plant pulp, there is in plant pulp content is there, the plant pulp content there is in paper industry. Is there. So here there is a link is there, the cellulose plant, okay, cellulose is produced by the plants. So whatever the paper industry is there, the paper industry collecting the plants. That industry to produce paper using the plant pulp. Okay, see, whatever the plant produced cellulose content is there, that content is called as pulp content. That pulp content used by the paper industries and produces the paper. So that is about that paper industry. So sir, can you give me example one paper? Not paper, pulp producing plant. We know about the bamboo. Bamboo is one of the plant. Okay, the bamboo is one of the plant, right? That plant produces the pulp. Okay, that one is called as bamboo pulp. That pulp we are using within paper industry to produce the paper, to make the paper, right? So that is about. See, I like to say here one of the points. So here in your classroom, two friends are there. Both the friends are writing notes, right? One of your friend having classmate notebook, and other one having somewhat rough note kind of book. And if we compare the quality of the two books papers, if we compare the paper quality of the paper, okay, the two book papers, in that case one is very pure white color, other one is that somewhat not pure white. So why the paper color is varied? Both are having cellulose. Remember, the percentage of cellulose is altered. The percentage of cellulose is altered. See, one is the high quality cellulose, other one is the low quality cellulose. That is one of the factor. And other one is percentage of the cellulose is also important factor. High percentage of the cellulose and the low percentage of the cellulose. So that's the reason why the color of the paper is altered, right? The quality of the paper is also altered. The quality of the paper also altered. See, that is about that the percentage of cellulose and the color of the paper. Color of the paper, right? So, see, within the laboratory also we are using some of the cellulose papers which are called as Wattman #1 filter papers. Wattman #1 filter papers. These are highly recommended for the biochemical laboratory. These are highly recommended for the biochemical laboratory, which are using for the separation of different types of pigments and the filtration process also. So here, this is about that cellulose. So cellulose, these are the major points. All of you remember: tunica, animals, right, and the plant pulp content, paper industry. And the important point, the cellulose not react with iodine. The cellulose, not react with iodine solution. See, this point is very easy. If we know we studied in the 10th class or 9th class, maybe 7th class, if the starch interact with that iodine solution, blue color complex formation takes place. That time we are given one statement: carbohydrates treated with the iodine solution, blue color complex will be formed. That's the reason why the green color leaf turns into the blue color complex structure. Here, in any part cellulose is present. If the cellulose part is interact with the whatever that iodine solution is there, right? So in between the iodine and the cellulose, there is no reaction. So why the reaction is absent between these two? Why? Sir, you are saying carbohydrates. Okay, definitely. Cellulose is also carbohydrate, no doubt. And the starch is also carbohydrate. Starch have the capacity to interact with or to react with iodine solution. It forms the blue color complex. Why not the cellulose? Cellulose is not react with iodine. Cellulose is not react with the iodine. Why? Why it's not forms any color complex? Remember, there is no reaction between iodine solution and the cellulose. Okay, here there is no color complex. If the reaction takes place, definitely there is a color complex formation takes place. Between the cellulose and the iodine, there is no reaction, there is no color complex formation. Remember, if starch is taken, in that case definitely reaction takes place. Definitely there is a blue color complex formation takes place. All of the students better search for this answer, otherwise tomorrow class I will explain why between these two no reaction, why that the cellulose and why the starch and the iodine the reaction takes place. Okay? That is about the very first Homo Polysaccharide, cellulose. Next class we will discuss about remaining all Homo Polysaccharides.